Sushruta for NEET is now available on the Google Play Store. Try it out for free.
Hi, my dear students. I am Professor G.K Bhat. Hearty welcome to our JnanaMarga.in biology classes. Having studied the chapters of 11th standard, digestion and absorption, and breathing and exchange of gases, it is the right time for us to study body fluids and circulation. You know, whatever we eat, whatever the food substances that get digested in our alimentary canal, ultimately, you should reach each and every cell. And in each and every cell, the food that is absorbed has to made use to get the energy. Now the question is, how the digested food, absorbed food from the alimentary canal reaches each and every cell? The answer is, we have two fluids for that. One is the blood and another one is the lymph. These two body fluids and their circulation enables each and every cell to get the food and thereby to get the energy. So, in today's class, we shall study the details of body fluids and circulation. Right? And these are the subtopics that we are going to study under this particular chapter. All living cells have to be provided with the nutrients. That's what I just said. Not only that, they should be provided with the oxygen. For what purpose? To break down the nutrients, whatever that the cells get, and thereby to get to the energy. No doubt all living cells should get other essential substances also, especially hormones. And also the waste products which are harmful to our body. When they are produced, then in that itself, they have to be removed continuously for the healthy functioning of the body, for the healthy functioning of the tissues of the body. It is therefore very essential to have efficient mechanism of the movement of these substances to the cells and from the cells. If we look into this mechanism, it is different for different animals. Different animals have evolved different methods of transport. Transport of these food substances, transport of these vital gases like oxygen and carbon dioxide, transport of the excretory material, transport of the hormones. Now let us have a survey. Let us start with the simplest animals to the most complex organism. Simple organisms like sponges and coelenterates circulate water from their surroundings through their body cavities in order to facilitate each and every cell to have exchange of these substances. Look into the sponges. In sponges, there are ostia. Through these small apertures, ostia, the water containing the food particles containing the dissolved oxygen enters into the cavity of the sponge called spongocoel. Inside this spongocoel, the food particles are digested by the cells concerned in that what is called intracellular digestion. After that, whatever the food that is not digested, it comes out to the osculum, the aperture which is meant for that. At the same time, the water which has the dissolved oxygen in it, is exchanged with the cells there, lining the spongocoel. In turn, dissolved carbon dioxide comes out to the spongocoel, and outgoing water to the osculum carries out this dissolved carbon dioxide also. So in this way, water which enters into the ostia, it supplies the food materials and the dissolved oxygen to the cells of the sponge body, and outgoing water carries this undigested food. Along with that, it also carries the dissolved carbon dioxide. At the same time, the outgoing water from the spongocoel to the outside to the osculum also carries nitrogenous waste in the form of ammonia. So that also is passed out. So in this way, these substances which are essential by the body, which are essential for the animal, they are taken in, utilized, and whatever that is unwanted, they are passed out to the body, out from the body to the osculum. If this is the scenario with regard to the sponges, in case of ciliates like Hydra, Adamsia, in all these, there is only one aperture, the mouth, which helps both as a mouth and anus. Through the mouth, water enters in, bringing the food particles, bringing the dissolved oxygen, and while going out to the mouth, it takes away the undigested food, it takes away the dissolved carbon dioxide, it takes away the nitrogen's waste in the form of ammonia, just like whatever you have seen in case of sponges. So these are the two things which you have observed in the simplest organisms like sponges and ciliates. Now, with regard to the more complex organisms, we come across special fluids to do this function. And these special fluids enable the transport of the materials within the body. Which are these special fluids? As I just mentioned at the beginning of the class, one is the blood. Blood is the most commonly used body fluid by most of the higher organisms, including we human beings. And another body fluid is the lymph. Lymph also helps in the transport of certain substances, especially the fatty acids and glycerol. So, in this chapter for body fluids and its circulation, we are going to study more about this composition and properties of the blood and the lymph, which is otherwise called a tissue fluid. And also, we are going to study about the mechanism of circulation for blood. Let us start the details. Look into this diagram. You don't see this diagram in the NCERT book. Most of the time, you think that each and every cell of our body is supplied with the nutrients, is supplied with the dissolved oxygen or other substances. But it is not true. Look at the diagram here. Whatever you see there in pink color, it is a tissue, mass of cells. What you see outer to this, which is shown in yellow color, this is what you have, some fluid surrounding this tissue, and this fluid is called, this fluid which is present outer to the tissue is called tissue fluid. It is sometimes also called interstitial fluid, also called the extracellular fluid. How this fluid is formed? Look here. We have a blood capillary bringing the blood to the tissues. Whenever this blood capillary brings the blood to the tissue, from the capillary, much of the water content or much of the content of this blood capillary, with the exception of the red blood corpuscles, white blood corpuscles, blood platelets, and larger proteins, with the exception of all these, whenever I say, with the exception of formed elements, that means the remaining is a plasma. From the plasma, so whatever proteins are there in that plasma, except that, you see that all other fluid component, it moves out into the space surrounding the tissue, and this is known as a tissue fluid. So, what tissue fluid you have? It is nothing but plasma filtered out of capillary. It is the plasma that goes out, keeping away the foreign elements, whatever you have already seen, keeping away the larger proteins. Now, the plasma moves out and gathers, accumulates around this tissue, and this is called tissue fluid. Whenever this food material comes there, say glucose, this glucose from the capillary, it now passes to tissue fluid. From the tissue fluid, the glucose passes to the tissue. It passes to the tissue. In the same way, whenever the cells need oxygen, the oxygen also from the blood capillary, first it comes to tissue fluid. From the tissue fluid, it enters into the cells. When urea is found, urea that is found in the cells, from the tissue, the urea comes first to the tissue fluid. From the tissue fluid, it enters into the blood capillary. Now, it is the blood capillary which takes the blood away from the tissue. Now we call it a venous venule. It comes to the venule here. At the same time, the carbon dioxide which is produced in the cell due to internal cellular respiration, that carbon dioxide also comes out from the tissue to the tissue fluid. From there it enters into the venules. And this venule, which forms vein, it takes this away from the tissues to take it further towards the heart. So this is what is happening at the tissue level. And later, from some of this tissue fluid, whatever you come across, whatever you see around the tissue, they enter into a special type of vessel called the lymphatic vessels. The moment they enter into the lymphatic vessel, tissue fluid, some of the tissue fluid, the moment they enter into the lymphatic vessel, now they are no more called a tissue fluid, but they are called by another term, what is known as lymph. In fact, lymph is nothing but tissue fluid. Lymph is nothing but a tissue fluid, but it gets the name lymph once this tissue fluid enters into the lymphatic vessel. Now you may get the doubt. From here, where they go? From the lymphatic vessel, or the lymph inside the lymphatic vessel is taken further, and on either side of the neck region, they join the subclavian vein. They join a blood vessel called subclavian vein. Therefore, remember, the tissue fluid is a formula for blood, and the tissue fluid later joins the blood. There's no loss to the body at all, because the fluid that comes out from the blood vessel, the fluid that comes out from the blood capillary, since it is going to form the tissue fluid, Don't think that there is loss of that much of fluid from the blood, because it is returned back to the blood. So this point you have to know before studying the further aspect of this particular chapter. Right? Now let us look into the details of this. Blood. While studying these animal tissues, you have heard under the connective tissue, special type of connective tissue. The special type of connective tissue included bone, cartilage, and blood. You have studied about the bone and cartilage earlier. Now we are going to look into the blood. Blood is a special connective tissue consisting of two parts. One is what is called fluid matrix plasma. Another one is the formed elements. These are the two components of the blood. Plasma part and embedded in the plasma, the formed elements. How much of the blood constitutes plasma? How much of the blood constitutes the formed elements? Let us start to look into the plasma. In fact, 55% of the blood is formed of plasma. 45% is formed of the formed elements. Plasma is a light yellow colored, slight yellow colored, what you call a straw colored fluid, which is viscous, constituting nearly 55% of the blood. Important. 55% of the blood is formed of plasma. In the plasma, 90 to 92% is water. 90 to 92% of the plasma is water, and 6 to 8% is a protein. I repeat, in the plasma, 90 to 92% is water and 6 to 8% is protein. When we speak about the protein, there are three types of proteins. Which are they? Fibrinogen, globulin, and albumins. These are the three types of major proteins found in the plasma. Major types of proteins. Each with its own function to perform. Fibrinogens are needed for clotting or coagulation of the blood. Globulins are primarily involved in immune response, that is the defense mechanism of the body, and albumins help in maintenance of osmotic balance. Always ask a question, which are the proteins found in plasma and what is their function? Or you may be given a column, matching the column question with the column 1, column 2. Column 1, these are different types of proteins, and column 2, different functions. Easy to remember. Fibrinogen is going to form fibrin threads whenever you get to the wound and all. Fibrinogens are needed for clotting of the blood or coagulation of the blood. Immunoglobulins or globulins are involved in the defense mechanism, and albumins are concerned with the osmotic balance. Is it that these are the only things that will be present in plasma? Answer is no. Plasma also contains a small amounts of minerals like sodium, calcium, magnesium, bicarbonates, and chlorates, etc. You have to remember this. Calcium, magnesium, and bicarbonates, sodium, chloride. These are also present in the plasma. Apart from that, whenever the blood moves from one place to another place, one part of the body to another part of the body, during this transit, we also come across in the blood glucose, amino acids, and lipids. So, apart from whatever you have seen in the plasma, water, proteins, fibrinogen, globulin, and this albumin, you also come across sodium, chloride, calcium, magnesium, bicarbonates. Along with that, whenever the blood is flowing from one part to another part, whenever it receives some other substances like glucose, amino acids, and lipids, that it also carries from one part to another part. And these glucose, amino acids, and lipids are present as they're always in a transit inside the body or any body. And apart from that, there are many factors of coagulation or factors for coagulation or clotting. These factors are also present in the plasma in an inactive form. So this is what you see in the plasma. Now let us see which are these factors for coagulation. Again, this information is not there in your NCERT book, but you are supposed to know at least the 4 of them are very important. The first 4, which you know, which you are supposed to know very well. What are these factors? What are these coagulation factors? These coagulation factors are always written in Roman letters. Factor 1, factor 2, factor 3, factor 4, up to factor 13. Factor 1 is the fibrinogen. Factor 2 is the prothrombin. Factor 3 is the thromboplastin. Factor 4 is calcium. These 4 factors are very much concerned with the coagulation of the blood whenever we get some wound in our body. Remember, factor 1 is fibrinogen. Whenever we say, you know already, whenever we add this suffix gen or prefix pro, means they are in inactive form. You have factor 1 fibrinogen, factor 2 prothrombin. Fibrinogen is an inactive form of a fibrin. Prothrombin is an inactive form of a thrombin. And you have factor 3 thromboplastin. Thromboplastin is also called thrombokinase. Thrombokinase. It is an enzyme concerned with the blood clotting. And you have factor 4 calcium. These 4, which factor and what is its name, very, very important. Apart from that, you need not give much importance to this factor 5 or factor 6, but you must know which is the factor 8 and factor 9, which are directly concerned with the hemophilia. Factor 3 is known as anti-hemophilic factor. Factor 9 is known as the Christmas factor or plasma thromboplastin component. These you must know, and let us discuss about that later. Factor 8 and factor 9. One is responsible for hemophilia A and another is responsible for hemophilia B. Right? Rest of the thing, just have knowledge of that. If at all it is asked sometime in a NEET examination, you must be in a position to just get that point. But at this time, I am concentrating on factor 1, factor 2, factor 3, factor 4. The most important coagulating factors, coagulation factors. Okay. Now, you must also know that plasma without this clotting factor is simply called serum. Many times question will be like this: what is serum? Plasma without clotting factors. The clotting factors are the one which I have just mentioned. This is what we have serum. Now let us look into formed elements. We have already heard that 45% of the blood is formed for formed elements. What are the components of the formed elements? Erythrocytes are red blood corpuscles, leukocytes are white blood corpuscles, and platelets or thromboplastin. These are platelets. These are the altogether they are known as the formed elements. Erythrocytes, leukocytes, and blood platelets. Blood platelets. These together they are known as the formed elements. Let us study these one by one. Look here. Red blood corpuscles, blood platelets, thrombocytes. Here you have eosinophils, basophils, neutrophils, monocytes, and lymphocytes. All these are white blood corpuscles. And it is needed. You must be in a position to identify them if the diagram is given like this. I shall tell you how to identify. Especially these. Which is the eosinophil, which is the basophil, which is the neutrophil, which is the monocyte, which is the lymphocytes. Easily can make out. In a neutrophil, the nucleus is lobed. You can clearly see 3 to 4 lobed. If the nucleus is 3 to 4 lobed, it is neutrophil. If it is somewhat bilobed with 2 lobes, eosinophil. If the nucleus is somewhat S-shaped, it is the basophil. If the nucleus is bean-shaped, it is monocyte. If the nucleus is somewhat spherical, bit large, T lymphocyte, small, B lymphocyte. So this is the picture of that of the different types of RBC, blood platelets, and WBCs. Let us start studying these one by one. First, take up erythrocytes. Erythrocytes, also known as red blood cells or red blood corpuscles, are the most abundant of all the cells in the blood. That means, in a healthy adult human, especially man, on an average, we come across 5 million to 5.5 million of red blood corpuscles per cubic millimeter of blood. Per cubic millimeter of blood, we come across 5 million to 5.5 million of red blood corpuscles in the adult man. When you say that it is a 5.2 to 5.5 million of red blood corpuscles per cubic millimeter in the adult human man, the next question arises, is it is less in woman? Yes. Around 0.5 million is less in adult woman. The difference is around 0.5 million. 0.5 million is more in case of adult man. Red blood corpuscles are always formed in what is known as the red bone marrow in the adult. General ask a question. Where the red blood cells are formed? 1, 2, 3, 4. And most of the time we write red bone marrow in the adult. But you must remember, the red blood corpuscles are formed in different parts of our human body during embryonic condition. Whenever the fetus develops inside our body, when the fetus develops in the womb of the mother, you see the hematopoiesis or red blood cells to form in different parts of the fetus. Look here, When the fetus is passing through the first month of the development, fetal month, 1 to 9 it is shown, the very first, in the first month of the fetal development, the hematopoiesis. Hematopoiesis is the formation of the blood cells. The hematopoiesis starts in the first month in the liver. Once it starts in the liver during the first month, it reaches the maximum number of formation of these blood cells somewhere during this 4th to 5th month time, and gradually it starts declining, and by the time the baby is born, liver stops producing these blood cells. Once the blood cell production starts in the liver during the first month, when the fetus passes through the second month and it advances towards third month, immediately, in between this second and third month, the blood cell formation starts to happen in the spleen. And in the spleen, it continues up to nearly 7th month, reaching the peak in between the 4th and 5th month. So remember, during both, in between the 4th and 5th month, it is the liver and the spleen. They are mainly concerned with the blood cells formation and not other parts of the body. Right? But at the same time, just near the 3rd month, you also see maximum amount of these blood cells are produced in the yolk sac. In the yolk sac. And then, after 4th month, now the blood cell formation starts in bone marrow, in the bone marrow of longer bones, and also it starts in the lymph nodes. Once it starts in the 4th month, later the production continues throughout the life of an individual in both the lymph nodes and in the bone marrow of the bones like vertebra and pelvis, sternum, ribs, and also in the tibia, in the tibial bones, and also the femur bones. So, after birth, the blood cell formation is more inside the bone marrow. Next to that is inside the lymph nodes. And these are the different areas where these blood cells are formed with the differential number or percentage. But what you have to remember, need not remember from what month to what month and all. No, you just remember, it is not that bone marrow alone is a place of production of blood cells. It is not that lymph nodes alone is a place of production of blood cells. So blood cells are produced during the embryonic condition in the liver, in the spleen, in the yolk sac. This is what you have to remember. So liver, spleen, yolk sac are the places of production of blood cells along with the bone marrow during the embryonic condition. We can't leave because right from the 4th month, right from the 4th or the 5th month, we see that the blood cells are produced inside the lymph node as well as inside the bone marrow. After the birth, no doubt it continues to the maximum number in only these 2 organs, these 2 regions, bone marrow and the lymph nodes. But prior to the birth, the the yolk sac, the spleen and the liver are also involved in the production of blood cells, but after birth, they are not involved. But the spleen is concerned very much. Spleen is always said it is the birthplace of the red blood cells and also death place. It is a cradle, no doubt, and also it is a graveyard. Okay, so this is a little bit additional information preparing you for the NEET exam. And one can have this screenshot to understand this concept better. Red blood corpuscles are devoid of nucleus in most of the mammals and are biconcave in shape. They have hemoglobin, that is why they are red colored. As you know, hemoglobin is an iron-containing complex protein. In our blood, generally, in a healthy individual, we come across 12 to 16 grams of hemoglobin in every 100 ml blood. 12 to 16 grams of hemoglobin in healthy adult man. Generally, around 2 grams of this hemoglobin percent is less in woman. In a healthy individual, in a healthy man, if it is 12 to 16 grams of hemoglobin for every 100 ml of blood, in a healthy woman, it may be around 10 to 12 or around 11 to 12, 13 like that. Generally, 2 grams of hemoglobin is less for every 100 ml of blood in case of a woman when compared to man. These hemoglobin molecules play very important, significant role in transport of respiratory gases. You must know that. Red blood corpuscles have an average lifespan of 120 days, after which they are destroyed in the spleen. That's why spleen is called graveyard of RBC. It is a cradle. You know that a cradle, a swing in which a baby is taken care of, put and taken care of. It is a cradle as well as a graveyard. It is the birthplace as well as the death place for the red blood corpuscles. Remember, for every 120 days, your red blood corpuscles get destroyed. Therefore, I always request my young friends, you people, please cultivate the habit of donating blood. Blood donation is the best service that you can do to the mankind. That's the best service that you can extend to the community. Of course, there is an age limit. Whenever you attain 18 years of age, or whenever you put up, along with the 18 year of your age, with 45 kg of body weight, you are well qualified to donate the blood. I repeat, a boy or girl who celebrates 18th birthday, who puts up with 45 kg of weight, becomes eligible for blood donation up to this age, up to attaining this particular weight. You are not supposed to donate the blood. But whenever I used to work in colleges and all, a good number of students who are studying in the first year or second year PU or 11th and 12th standard, they used to voluntarily come forward, telling that, sir, I might not have attained 18 years of age, but I'm already 45. I have already crossed that 45 kg of weight. Why don't you take my blood? Why don't you permit me to donate the blood? At that time, I used to allow them to just contact the doctors, and with the permission of the doctors, and after testing their hemoglobin percentage, always for blood donation, one should get the hemoglobin percentage tested. Less than 10 gram of hemoglobin, a person having less than 10 gram of hemoglobin is generally not entitled to donate the blood on that day. So, if at all the hemoglobin percentage is above 10, if at all your age is above 18, if at all your weight is above 45, in all aspects, you are the potential candidate to start the blood donation. My request is that please start celebrating your 18th birthday with the blood donation. Make it a point to go to another hospital and start that. And at least in every year, you start first one time, then you just increase it to 2 times. And after you get married and all, you can make it 3 times. One during your birthday, second during your better half's birthday, third during the wedding anniversary day. At least 3 times you start to donating blood. It's the donation of the blood is the best service that you can do to the society. I hope, because you know that there is no substitute to human blood. Whenever a human being needs blood, human blood only has to be given. We cannot give the blood of the buffalo or a pig or goat like that. And we will not be knowing when we require blood, when we may need the blood. We do not know when our relatives may need the blood. And we expect whenever we require the blood or whenever our relatives need the blood, we expect all others to come and to donate the blood. But when we are healthy, when we are able to donate blood, when we are capable of doing that, why not to do now? I hope my all students take this matter very seriously and start to donating blood. I myself have donated 34 times of blood to the needy people. I hope you will follow it. And I have organized a number of blood donation camps for the charitable hospitals and all. I hope you will take it as a very serious matter, very important matter in your life. Right. Now, this is the diagram showing you line diagrams, form elements. This is a lymphocyte, this is a monocyte, this is a neutrophil, this is a eosinophil, and this is a basophil. Right? Having studied about erythrocytes, now let us look into leukocytes. Leukocytes are also known as white blood cells or WBC, as they are colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which averages 6,000 to 8,000 per cubic millimeter of blood. You know, leukocytes are always called soldiers of the body. All the 24 hours, they'll be very much vigilant, taking care of our body, engulfing or destroying whatever that the microbes they try to enter into our body. Leukocytes are generally short-lived. We have two major categories, main categories of WBCs. One is granulocytes and another one agranulocytes. What are these granulocytes and agranulocytes? Granulocytes means the white blood corpuscles with the minute granules in their cytoplasm. Whereas agranulocytes are the leukocytes without any granules in the cytoplasm. Remember, granules in the cytoplasm. And these granules, they take a different stain when we do this differential staining of the leukocytes. Neutrophils, eosinophils or acidophils, and basophils are different types of granulocytes, while lymphocytes and monocytes are the agranulocytes. Granulocytes include neutrophils, eosinophils, also called acidophils, eosinophils, also called acidophils. And basophils are different types of granulocytes, whereas lymphocytes and monocytes are the agranulocytes. Now let us look into the percentage of these different white blood corpuscles. Amongst these 5 different types of white blood cells or leukocytes, the most abundant leukocyte is the neutrophil. Neutrophils are the most abundant cells, occurring to the level of 60 to 65%. And the least number of leukocytes are of basophils. Basophils are the least among the leukocytes with around 0.5 to 1%. Most abundant is the neutrophils, 60 to 65%. Next to this neutrophils, the most abundant white blood corpuscles is the lymphocytes. Next to neutrophils, lymphocytes, which occur 20 to 25%. Most abundant is the neutrophil. Next to that neutrophil, the most abundant is lymphocyte. Next to this lymphocyte, the most abundant is the monocytes, 6 to 8%. Next to this monocyte, the most abundant, next to that, is that of the acidophils or eosinophils. Eosinophils, 2 to 3%. So, according to their number, the leukocytes number, which is highest or from the highest to lowest, are neutrophils. Next to the neutrophils, lymphocytes. Next to the lymphocytes, it is the monocytes. Next to the monocytes, it is the eosinophils. Next to the eosinophils, the least one is the basophils. Basophils are the least one, 0.5 to 1. Next to basophil, it is the eosinophil, 2 to 3%. And next to eosinophil, more in number is that of the monocyte. It is 6 to 8%. Next to that is the lymphocyte. Lymphocyte is 20 to 25%. And the last and the most abundant is the neutrophils. This is with regard to the number. And what is the function of neutrophil? Neutrophils and monocytes are phagocytic in nature. They are the phagocytic cells which destroy foreign organisms entering the body. Which one? Neutrophils and monocytes. These are always phagocytic cells. Always engulf or destroy whatever that enters into our body. Then, the basophils, which are least in number, they secrete 3 important chemicals called histamine, serotonin, heparin, and are also involved in inflammatory reaction. Inflammatory reaction is the first response of the immune system. The first response of the immune system is called inflammatory reaction, which involves the edema of that particular part of the body, which is hurt, warmthness in that, redness in that, and pain. Edema, that is the swelling, pain, reddening of that part, and warmthening of that part. These are the 4 important signals of inflammatory reaction. And with this, the white blood corpus that is concerned is the basophil. The basophils also secrete histamine, serotonin, heparin. Histamine is vasodilator. It is always making the blood vessels to dilate. Serotonin is vasoconstrictor. It constricts the blood vessels. Heparin is anticoagulant. Histamine, vasodilator. Vaso refers to blood vessel. Vasodilator. Serotonin, vasoconstrictor. And heparin is anticoagulant. All these are secreted by basophils. Eosinophils resist infection, are also associated with the allergic reactions. And lymphocytes are of 2 types, B lymphocytes and T lymphocytes. These are mainly responsible for immune response of the body. You are going to study about that details later. B lymphocytes, which are concerned with the antibody-mediated immunity, and the T lymphocytes are concerned with the cell-mediated immunity. That we are going to study in detail later. So you must know what is the function of these neutrophils and that of the monocytes. What is the function of basophils? What is the function of eosinophils? What is the function of lymphocytes? Right? Now let us come to the platelets. Platelets are also called thrombocytes. And these are cell fragments produced from megakaryocyte. Mega means large. Karyocyte, which are found inside the bone marrow, special cells. Platelets, otherwise called thrombocytes, are produced inside the bone marrow. And generally, our blood contains 1,50,000 to 3,50,000 platelets per cubic millimeter blood. 1,50,000 to 3,50,000. Blood platelets can release a variety of substances, most of which are involved in coagulation or clotting of blood. I already told you about clotting of blood. Blood platelets are concerned with that. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. So blood platelets are mainly concerned with the clotting disorders. They are very much concerned with clotting of the blood. You can see here, this is a diagram showing you the neutrophil with many lobes there. That's the main point. Multi-lobed one. Neutrophils. And this is what you have eosinophil, bilobed, neutrophil, eosinophil with the two lobes. And this is somewhat S-shaped nucleus having a basophil, right? And this is what you have, the lymphocytes, right? Now, if you look further, this is the monocytes with the bean-shaped nucleus. Bean-shaped nucleus. Look here, granular lymphocyte. And if you look into this, now you must tell me, what are these? And look into that. Out of these, the largest one and the smallest one, you find it. This is, as you can easily make out, this is neutrophil. Look into this multi-lobed nucleus. Neutrophil. This is eosinophil or acidophil. This is basophil. This is lymphocyte. And these are monocytes. Monocytes are the largest cells with the 14 to 17 mm diameter. And lymphocyte is the smallest WBC in size. Monocyte is the largest one. Lymphocyte is the smallest one. Right? This is with regard to the size of the cells. And this is with regard to percentage. Already heard. This is the neutrophil with the highest number. Next to that highest in number is the lymphocyte. Next to that highest in the number is the monocyte. Next to that is the acidophil. Least one is that of the basophil. Right? Now, we have to start with these blood groups in our next class.